Hi dear friends, now this is the second video, sexual reproduction. Sexual reproductive parts of plant is known as flower. So the flower is specialized reproductive structure of a plant in which sexual reproduction takes place. In most of the angiosperm, that is flowering plants are there, so flower is the sexual reproductive parts. Then what is the parts of typical flower? It consists of mainly two parts, that is accessory whorl, that means it is not involved in reproduction. The main function is protection. There are two types, calyx, that is outermost whorl, that is made up of sepal, corolla, that is made up of petals. Then second word that is important one is essential whorls, that is involved in reproduction. That is made up of andrecium, male reproductive structure, gynecium, female reproductive structure. See the structure LS of flower, that is the stalk of the flower is known as pedicel. This is the stalk of the flower, and this is the receptacle or thalamus we can say. From here the floral whorls are arising. The outermost whorl is known as calyx, that is made up of sepals. This is the first whorl. Second whorl is corolla, that is made up of petals. Then third one is that is andrecium. This all of the andrecium made up of individual part is known as stamen. This is the stamen, and each stamen consists of that is anther and filament. This portion is anther, this one is the filament. Then the innermost whorl of the flower is known as female reproductive structure, that is gynecium or known as pistil, P-I-S-T-I-L, pistil. It consists of ovary, this is the ovary contain ovule inside, then style and stigma. So the parts of the flower, outermost whorl is calyx made up of sepal, second one is corolla made up of petal, third whorl is that is andrecium made up of stamen. Each stamen consists of anther and filament, and innermost whorl is that is gynecium. It is made up of ovary, style, stigma. Ovary contain ovule, and inside the ovule we can say the female gamete will be here, that is egg cell. See here again same thing, essential whorl andrecium, male reproductive structure made up of stamen. Each stamen consists of filament, connective, anther. Structure of anther, next we will see, that is structure of anther. See, if you take the TS of anther, that is anther is a this is anther, filament, connective and anther. Structure of anther, generally anther is dithecous, that is two lobe structure, and it is tetrasporangiate. Four that is lobes are present, that is tetrasporangious. Tetra means four. See the structure of anther, that is if you take the TS of anther, you can see the outermost whorl is known as epidermis. This is the epidermal layer made up of parenchyma cells. Second one is endothecium. See, this is endothecium, that is long elongated structure. Second one is endothecium, and below the endothecium that is middle layer. Middle layer is made up of 2-3 layers. Middle layers, epidermis, second endothecium and middle layers. Then this is the pink color structure, that is a tapetum. That is a nutritive tissue for the developing pollen grains here. That is the sporogenous tissue present inside the tapetum. So here sporogenous tissue, or they will produce the pollen grain, or microspore mother cells are present inside. Then the nutritive tissue tapetum, then middle layer endothecium, epidermis. Again once more, the structure outermost layer is epidermis, second one is endothecium, then middle layer, then the tapetum, that is the nutritive tissue. It can ask in the MCQ, what is the nutritive tissue inside the anther, that is the tapetum, and inside microspore mother cells. Normally then vein, that is the conductive tissue vein, and here that is mostly that is 16 spore mother cells will be present here inside the pollen sac. This we can say the pollen sac. Inside 16 spore mother cells are there. They will undergo meiosis when get maturity, and each spore mother cell will produce 4 haploid microspores or pollen grain. So in one pollen sac, 64 pollen grains you can see here, 64, 64, 64, 64 like that. So when they get maturity, when they produce the pollen grain inside, then this layer, that is the connective link here, this will break open and the pollen grains will come out. That is known as the dehiscence of anther. Anther will dehisce open and the pollen grains will come out. The TS of anther, once more the diagram. Epidermis, outermost protective layer, endothecium, subepidermal layer, middle layer made up of 3 layered structure, tapetum, innermost nutritive tissue, microspore mother cell, or it is known as sporogenous tissue. Microspore mother cell is diploid. It undergoes meiotic division and produces haploid pollen grains. It is otherwise known as microspores. See, these are dehiscence of anther loop. So this, when the pressure create here during the formation of pollen grain, this portion that is made up of parenchyma cells will break open and this pollen grains will come out. That is dehiscence of anther, opening of anther loop. Then this, when the anther dehisces, the pollen grains will be mostly at the time of dehiscence, it will be 2-celled stage. So here each microspore mother cell divides meiotically, just now I told, to form tetrad or haploid microspores, that is pollen grain. This is known as microsporogenesis. Microsporogenesis, genesis of microspores. Microspores means that is the pollen grain. Formation of pollen grain or development of microspores, that is known as microsporogenesis. And structure of microspore. Typical pollen grain is a non-motile haploid unicellular body with a single nucleus. But at the time of dehiscence, it will be 2 nucleated stage, and it consists of that is vegetative cell and a generative cell. And it is a double layered structure. Outer layer is known as exine. It may be smooth or spinular structure. And inner one is, that is intine. Inside, cytoplasm and nucleus are present. And this is the 2 nuclei, and it will form the 2 cell. One is the large vegetative cell, other one is the generative cell afterwards. And in between, that is thin layer, you can see, that is known as the germ pore. Through that is the indine will come out and form the pollen tube during germination. And that pollen grains at the time of dehiscence, it will be 2-celled stage. So that is tube nucleus or vegetative cell, and this is the generative cells. Beginning it will be unicellular, afterwards at the time of dehiscence it will be 2-celled stage. Tube nucleus or vegetative nucleus and generative cells. This generative cell undergoes mitotic division again, and they will form 2 male gametes. By the time the indine will come out through the germ pore and form tubular like structure, that is known as pollen tube. Pollen tube carry, that is the generative cells, and the tube cell will degenerate afterwards. Tube nucleus and generative cell carry by the pollen tube. Tube cell will degenerate, and generative cell again divides mitotically to form male gamete. And 2 male gamete along with this pollen tube is known as male gametophyte. So this is the male gametophyte generation. So that is from single cell nucleus it become double cell. At the time of dehiscence it will be double cell structure, tube nucleus and a generative cell or tube cell. And this tube cell will this generative cell divide into 2 male gametes, and tube nucleus and the generative cells will come out along with the indine and form the pollen tube, and this pollen tube along with the male gamete is known as male gametophyte. Male gametes form from generative cells. Development of male gametophyte. Pollen grain undergoes mitotic division and produce vegetative cell and generative cell. Vegetative cell is otherwise known as tube cell. Generative cell produce 2 male gametes. At the time of dispersal from anther, the pollen grains are in 2-celled stage. That is vegetative cell, tube cell and generative cell. That is pollen tube with the 2 male gametes are called male gametophyte. This is the development of male gametophyte from microspores or pollen grains. Then we'll come to the female reproductive structure next class. Okay, thank you.